Hello, welcome to Biology, the scientific study of life. More about Mendel's heritable factors. Morgan's experimental evidence, scientific inquiry. The first solid evidence associating a specific gene with a specific chromosome came from the work of Thomas Hunt Morgan, an experimental embryologist at Columbia University early in the 20th century. Although Morgan has initially skeptical about both Mendelism and the chromosome theory, his early experiments provided convincing evidence that chromosomes are indeed the location of Mendelism or Mendel's heritable factors. Morgan's choice of experimental organism. Many times in the history of biology, important discoveries have come to those insightful enough or lucky enough to choose an experimental organism suitable for the research problem being tackled. Mendel chose the garden bee because a number of distinct varieties were available. For his work, Morgan selected a species of a fruit fly, Drosophila melonsgaster, a common generally innocuous insect that feeds on the fungi growing on fruit. Fruit flies are prolific breeders. A single mating will produce hundreds of offspring, and a new generation can be bred every 2 weeks. These characteristics make the fruit fly a convenient organism for genetic studies. Morgan's laboratory soon became known as the fly room. Another advantage of the fruit fly is that it has only 4 pairs of chromosomes, which are easily distinguishable with a light microscope. There are 3 pairs of autosomes and one pair of 6 chromosomes. Female fruit flies have a homologous pair of X chromosomes, and male have 1 X chromosome and 1 U chromosome. While Mendel could readily obtain different P varieties, there were no convenient suppliers of fruit fly varieties for Morgan employer. Indeed, he has probably the first person to want different varieties of this common insect. After a year of breeding flies and looking for variant individuals, Morgan was rewarded with the discovery of a single male fly with white eyes. Notice that, this one. And also instead of the usual red, the normal phenotype for a character. The phenotype most common in natural population, such as red eyes in Drosophila, is called a white type. Notice this type, red eye, and this one, white eye. Boom. Morgan and his student invented a notation for symbolizing alleles in Drosophila that is still widely used for fruit flies. For a given character in flies, the gene takes its symbol from the first mutant, non-white type, which means discovered. Thus, the allele for white eyes in Drosophila is symbolized by W. Notice. Okay, and superscript plus identifies the allele for the wild type trait W positive for the allele for red eyes. For example, over the years, different gene notation systems have been developed for different organisms. For example, human genes are usually written in all capitals, such as HD, for instance, for the allele for Huntington's disease. Morgan's notice more about the Morgan's fruit Morgan's first mutants. White type Drosophila flies have red eyes, left, this one, Among his flies, Morgan discovered a mutant male with white eyes, this one. This variation made it possible for Morgan to trace a gene for eye color to specific chromosome in this inquiry. Correlation behavior of a gene's alleles with behavior of a chromosome pair. Morgan mated his white-eyed male fly with a red-eyed female. All the F1 offspring had red eyes, suggesting that the wild-type allele is dominant. When Morgan bred the F1 flies to each other, he observed the classical 3-to-1 phenotype ratio among the F2 offspring. However, there was a surprising additional result. The white eye trait showed up only in males. All the F2 females had red eyes, while half, notice that, notice while half the males had red eyes and half had white eyes. Therefore, Morgan concluded that somehow a fly's eye color was linked to its sex. If the eye color gene were unrelated to gender, one would have expected half of the white-eyed flies to be male and half female. A female fly has two X chromosome XX, while a male fly has an X and a Y XY. The correlation between the trait of white eye color and the male sex of the affected F2 flies suggested to Morgan that the gene affected in his white-eyed mutant was located exclusively on the X chromosome with no corresponding allele present on the Y chromosome. His reasoning can be followed in this inquiry. For a male, single copy of the mutant allele would confer white eyes. Since a male has only one X chromosome, there can be no white type allele W positive present to offset the recessive allele. On the other hand, a female could have white eye and both her sex chromosome. Notice that. Also could have female could have white eyes only in both her X chromosome carried the recessive mutant allele W. This was impossible for the F2 females in Morgan's experiment because all the F1 fathers had red eyes. Morgan's finding of the correlation between a particular trait and an individual's sex provided support for the chromosome theory of inheritance, namely that a specific gene is carried on a specific chromosome, in this case the eye color gene on the X chromosome. In addition, Morgan's work indicated that genes located on a sex chromosome exhibit unique inheritance patterns, which recognizing the importance of Morgan's early work, many bright students were attracted to his fly room. Back to inquiry. In a cross between a white-type female fruit fly and mutant white-eyed male, what color eyes will the F1 and F2 offspring have? Experiment. Morgan mated a white-type red-eyed female with a mutant white-eyed male. The F1 offspring all had red eyes. This is the generation F generation. Morgan then bred an F1 red-eyed female to an F1 red-eyed male to produce the F2 generation. The result is, so notice the result here. The F2 generation showed a typical Mendelian 3-to-1 ratio of red eyes to white eyes. However, no females displayed the white eye trait. They all had red eyes. Half the males had white eyes and half had red eyes. Generation 2. Conclusion. Notice the conclusion here also. Since all of if one offspring had red eyes, the mutant white eyes trait W must be recessive to the wild type red eye trait W positive, since the recessive trait white eyes was expressed only in males in the F2 generation. Morgan hypothesized that the eye color gene is located on the X chromosome and that there is no corresponding locus on the Y chromosome as diagrammed here. P generation, notice that closer look, and F generation, notice also W positive and F2 generation. Okay, thanks for listening.